Hello, everyone. Thank you very much for the invitation to speak to you today about the mechanisms of developing allergy, and particularly food allergy. Now, IgE-mediated food allergy, the underlying immune cells and molecules are fairly well known that are involved in the food allergy response. So following ingestion and digestion of the foods in the intestines, these antigens or allergens are presented by dendritic cells to naive T cells, and usually you should get a regulatory cell response. So these food antigens should be well tolerated by the immune system. But for some reason, there's a polarization towards a Th2 response, characterized by cytokines such as IL-4, IL-5, and IL-13, that results in B cell secretion of IgE. And then this IgE travels and is bound by tissue cells such as mast cells. And the next time that you eat this food, you will get an allergic response. I think recently what we're becoming more appreciative of is the role of other barriers in influencing this Th2 polarization. So for example, inflammatory responses within the skin can lead to the secretion of alarmins such as IL-33, 25, and TSLP, which then polarize ILC2s and DC2s into a Th2 phenotype, and these seem to assist in the induction of these Th2 lymphocytes. So this has led to the dual exposure hypothesis, whereby if you have exposure to a food antigen via another barrier site, such as the skin, before you see it in the gut, then you will have a higher risk of developing an allergic response to that food allergen when you do consume it. One of the most important components that we look at in the skin is the presence of microbes and the composition of microbes within the skin. So the skin microbiota is different at different skin sites. So for example, moist and sebum-rich skin has a different composition to dry skin. The hair follicles are very important because within the hair follicles, that seems to be where the Langerhans cells and the dendritic cells are able to directly sample microbes. Microbes can activate pattern recognition receptors. For example, toll-like receptor 2 is activated by the lipoteichoic acid from Staph epidermidis, but also released metabolites can activate the immune system. So the mucosal associated invariant T cells respond to riboflavin metabolites coming from microbes on the skin. And on the skin, it is quite a competitive place for a microbe to hang out. So microbes secrete antimicrobial factors such as bacteriocins, which kill other microbes. And keratinocytes also secrete a wide range of antimicrobial molecules, such as defensins and catalycidins. So within normal healthy skin, there should be an effective barrier, and there should be a homeostatic relationship between the microbes on the skin and the immune system. However, when dysbiosis occurs, as is often seen in inflamed skin, microbes such as Staphylococcus aureus overgrow. And Staph aureus secretes a number of proteases, toxins, superantigens, which will activate the immune system, which will disrupt the barrier, and which will facilitate antigen sampling through the skin, such as food antigens. And this could be one of the ways by which children with atopic dermatitis have a much higher risk of developing food allergy. And we could see this in a study that we did in South Africa a number of years ago. And what I'm plotting you here is the age and months of diagnosis of atopic dermatitis, food allergy, or allergic rhinitis. And as you can see, atopic dermatitis comes first, next comes food allergy, and next comes allergic rhinitis. And if you continue this on till a little bit older, then you'll also see asthma coming in here. So this is the atopic march, and it seems to start with this disrupted skin barrier very early in life. And if we look at sensitizations in these children, so we had kids from both rural and urban environments, and we've divided them here into those with atopic dermatitis or healthy controls for both environments. If we look at total IgE levels in these children, you can see in the kids with atopic dermatitis, they have much higher levels of IgE compared to their healthy controls from both environments. And surprisingly, if we look at food allergen specific IgE, in this case, I'm showing you egg, but it was similar for many of the other food allergens we looked at. You can see a much higher rate of sensitization to these food allergens in children with atopic dermatitis. Again, showing the importance of an intact skin barrier. And it wasn't just food allergens. If we look at aero allergens, so this is the house dust mite specific IgE. Again, you can see this huge increase in specific IgE to house dust mite in those kids with a disrupted skin barrier. But one of the surprising things that we saw was that when we looked at circulating cytokines, we looked at about 50 different cytokines. Some were associated with atopic dermatitis, like TARC and IL-16. So these are well-described atopic dermatitis associated cytokines. But the majority of the cytokines were not related to the disease status of the child, so whether they had atopic dermatitis or not, but were actually related to the living environment that they were in, so rural versus urban environment. And I've just shown you a couple of examples here. So rural kids, whether they have atopic dermatitis or are healthy, have higher circulating levels of IL-6 compared to urban children. Also, the rural children have higher circulating levels of IL-8 compared to urban children, whereas the opposite is true for IL-22. This is higher in the urban kids, and IL-15 is higher in the urban kids. So overall, while the disease is important for sensitization, the overall immune tone of the children seems to be very heavily influenced by environmental exposures. And this is a summary or graphical abstract of that particular paper. So it was 152 kids. What we did find from a atopic dermatitis perspective was increased in cytokines that are Th2 atopic dermatitis associated. We saw less of a Th17 response, which was interesting, but an awful lot of sensitization. So total IgE and specific IgEs were significantly increased in the AD population. But when it comes to most of the other immune markers we looked at, these were influenced by environment. And some of the major influences that we were able to tie into this was exposure to animals, both pets in the home, but also farm animals, the living environment, and exposure to smoke in the home, whether it be smoking, cigarette smoking, or heating fires within the home and cooking fires within the home. So we wanted to go a little bit deeper into looking at what were the immune mechanisms that were underpinning these really dramatic environmental effects on immune development in these kids. So we isolated peripheral blood mononuclear cells from these kids and performed RNA sequencing. And what you can see here, and again, this was a big surprise for us, a pleasant surprise, is we see three groupings when you look at the differentially expressed genes. And when you plotted these out on UMAP plots, what you see is all the urban kids, they all group together. And then we see two groupings for the rural children. So this group is very far away from the urban kids. And then you have an intermediate group. So this, we think, is a rural group that's transitioning towards a more urban type signature in immune development. And we've mixed in here the kids with atopic dermatitis with the healthy controls. Atopic dermatitis modified about 100 genes, and important genes like IgE, the heavy chain for IgE, was higher in the atopic dermatitis kids. But when you look at the number of genes that are different depending on environment, you can see, and so I should say, in our labeling, we've labeled this rural 1 and this is rural 2 in the next few slides. So the rural 1 group versus the urban group, there was 3,400 genes that were significantly different. 2,000 upregulated, 1,300 downregulated. The rural 2 group was a little bit closer to the urban population, so there was 2,600 genes that were different. And many of the genes were shared between the two rural groups in comparison to the urban group. So comparatively speaking, atopic dermatitis results in a change in about 100, 110 genes, whereas environment is driving thousands of genes to be significantly different in these immune cells from urban and rural environments. And when we look at the pathways associated with these changes, you will recognize a lot of these pathways. So the MAP kinase signaling, TNF signaling, chemokine signaling, Th17, a lot of the innate signaling such as toll-like receptors, and also Th1 and Th2 cell differentiation. And I just plotted out here some of the groupings of genes that I'd be most interested in. So, and this is plotting p-value versus log fold change. And if you look at chemokines, you can see nearly all of the chemokines are upregulated in the rural kids compared to the urban kids. So, excuse me, suggesting a higher level of immune activation in the rural kids. I'll talk more about G protein coupled receptors in a moment, but these are a family of transmembrane receptors. We have about 700 in humans, um, and many of these are involved in immunoregulatory processes. And some of the ones here that were upregulated in the rural kids, like GPR132, that's been shown to be really important for control of autoimmune responses and can be activated by microbial metabolites. GPR183 has been shown to have a protective role in SLE. GPR84 regulates Th2 cell effector function. GPR31 is essential for the induction of oral tolerance by maintaining IL-10 producing Tregs in the gut. And GPR15 regulates the preferential homing of FOXP3 cells to the gut. So all of these are upregulated in the rural kids, really suggesting that even though there's more immune activation, it's a very regulated type of immune activation. And then when we look at cytokines, what we see is, again, very important regulatory cytokines like IL-10 are increased in the rural kids compared to the urban kids, whereas cytokines and receptors associated with Th2 and Th17 responses are upregulated in the urban children. You'll see the IL-5 receptor and the IL-4 receptor in there, and also components of Th17 signaling. So a very, very, very different immune profile dependent on the living environment of the child. And then lastly, we did a co-expression network analysis, and we could identify 6 communities based on genes that were co-expressed. And these identified the urban, the rural, the 2 rural subpopulations. And overall, what I would say is that the immune cell profile of the urban kids in this analysis, this is suggestive of some kind of a metabolic compromise in these cells, actually. These immune cells don't seem to be metabolically as capable as the immune cells from kids living in a rural environment. And if you look into rural networks that are our signatures, you can see again a lot of immune cell signaling and immune cell activation and T cell activation pathways that characterize kids growing up in rural environments. And what were the exposures that most significantly associated with these community patterns? So animal exposure, whether it be pets or farm animals, whether the child was exposed or the mother was exposed during pregnancy. Exposure to sunlight was highly correlated with these patterns. What we asked here was the amount of time that the kids spent outdoors in the sun. So I think rather than a measure of directly the influence of sunlight, I think this is a measure of time spent outdoors in biodiverse environments. Delivery mode had it's significant, but a minor effect. And then the cooking and heating fuels were also associated with some of these changes. For example, having an open fire in the home, there was a higher expression level of the aryl hydrocarbon receptor and the associated pathways with that. So to summarize this, I think while atopic dermatitis is important for sensitization, actually the living environment is incredibly important for the polarization, the activation, and the regulation of the immune system. And I've listed here some of the most important factors. So exposure to pets, farm animals, cooking and heating fuels, time spent outdoors, and also socioeconomic factors are very important in determining what type of immune profile the child is expressing. So how are these exposures actually mediating their effects on the immune system? So we would suggest that actually these exposures, like exposures to animals or time spent outdoors, would change your exposure to microbes. And microbes can influence the immune system via many different ways, but the two ways we look at it are that there are direct effects of microbes on immune cells, excuse me, for example, via pattern recognition receptors, but perhaps more importantly are the secreted metabolites that can have a number of immune regulatory effects distant to the site that the bacteria is sitting, because these metabolites can often be absorbed and can have effects on other parts of the body. And we have many examples to show that this is true. So this is a study that we did a few years ago, and we were able to correlate, for example, specific microbial species shown here on the right with circulating cytokines. And this was in adults. So for example, the sulfa Vibrio correlate very well with Th1 cytokines like interferon gamma and IL-2. The halobacteria correlate really well with a Th2 profile, like with IL-4 and IL-13. Whereas Doria correlate really well with a Th17 response. So like with IL-17A and MIP1 alpha, MIP1 beta. So the type of um, polarization systemically is related to the composition of your gut microbiota. And also early in life, you remember we start life sterile and we acquire microbes over the first 2 years of life, and we acquire microbes from mother. That's vertical transmission at birth, but then horizontal transmission from siblings, from a wider social network, from animals happens a little bit later. Um, as these microbes are developing, they need to be supported. So there are dietary supports that come in, but also exposure to infection, which will require antibiotics, will have a detrimental effect on the development of this early life microbiota. So recently, we just published a study a few weeks ago. We looked at kids who were born during the pandemic enforced social restrictions to see if we could measure a difference in the acquisition and the development of the early life microbiome with reduced exposures to other people. And actually, we did see a difference. And what we could see was, and it wasn't an easy story to explain, because even though these kids had reduced exposures to people, they also had reduced use of antibiotics. Only 17% of the kids needed an antibiotic by 1 year of age. It's usually 80, 90% in outside of the pandemic associated restrictions. But we were also able to see that breastfeeding had a huge effect. Plant-based weaning foods had a very surprising effect. I'll show you some details of that in a moment. Siblings seem to be a very important source of protective microbes. Living in a rural environment, again, was heavily associated with the acquisition of microbes at 6 and 12 months of age. Having pets in the home had a very significant effect, in particularly the spore forming type of microbes. So these are ones that are not usually vertically transmitted, but pets seem to be an important source of these in the early life microbiome. And if we look at where a lot of these spore forming bacteria, such as the class of Clostridia, are coming from, what we calculated here was an exposure index. So we added 1 or 2 in the number scale, depending on the children's exposure during the first year of life to what's called an essential worker. So that's someone who was allowed to work outside the home, how many siblings the child had, number of adults in the household, if they were able to attend daycare or not outside the home, having pets in the home, living in a house compared to an apartment. So the bigger the number, the more of these exposures that they have. And what you can see very nicely is all of these types of Clostridia, they all associate very well with these exposures. So indeed, the exposures to environmental factors such as other people, such as animals, such as a rural environment, and reduced rates of infection and consequently antibiotics had a very profound effect on the acquisition of these early life microbes. And there was a really nice correlation with food allergy and atopic dermatitis in these children. So what I'm showing you here is at 12 months of age, sorry, at 6 months of age, the association of different microbes with atopic dermatitis at 1 or 2 years of age, having a skin prick test positive response to a food antigen, or a non-IgE mediated food reaction within the first 2 years of life. What stands out here are bifidobacteria. So these are negatively correlated with the risk of atopic dermatitis or sensitization to food allergens. So the more bifidobacteria you have, the more protected a child seems to be against developing both food allergy and atopic dermatitis. So we can take another step and say, okay, so you've got all these microbes, how are they actually exerting a beneficial effect on the immune system? And this is where I think I can bring in immunonutrition. So this is, in its broadest sense, relates to the direct and indirect effects of dietary components on the immune system development and functionality and responsiveness. Now, its direct and indirect effects is included in this definition. And we do know, of course, and all of you know this probably even better than I do, that the long-term dietary habits do influence chronic mediated disorders and response to infection. And nutrient excess or calorie excess may be equally detrimental to the immune system as deficiencies. And I think what's important for today is to realize that many of the dietary effects that we can measure on the immune system may actually be indirect effects because they're mediated via the microbiota, metabolism of these dietary factors. And I'll give you an example from Purina Venter's study a couple of years ago, where she looked at the risk of atopic dermatitis, asthma, and wheeze development. And this is child's age here on the x-axis and the risk on the y-axis. In children who have a filaggrin mutation, so they have a defective skin barrier, they're known to have a higher risk of developing atopic sensitization. And she compared them to children who didn't have a filaggrin mutation, but who were consuming a allergy prevention diet or a non-allergy prevention diet. And if we look here at atopic dermatitis, so the top line is in those kids who have the filaggrin mutation, so they have the highest risk of atopic dermatitis. And then if you look at these two lines, you can see the kids who were on an allergy protective diet had a much lower level of atopic dermatitis. But those kids who didn't have a filaggrin mutation but were on a non-allergy protective diet, they actually had a much more similar rate of atopic dermatitis as those with a mutation that predisposes. So the reason I put this in here is just to give you an idea of how important diet is in protecting against allergic diseases such as AD and food allergy. And relative to a genetic risk factor, it's nearly as important. So in the cohort of children that I showed you a few minutes ago, that the early life microbiome was heavily influenced by their exposures early in life. We also measured the introduction of complementary foods in the 6 to 12 month time frame, and I was really surprised when we generated this heat map that so many foods actually either negatively or positively correlate with specific microbes within the gut. And so, for example, Akkermansia was positively correlated with foods like pistachio and macadamia. Bifidobacterium, and remember, this is the type of microbes that seem to be very protective against AD and food allergy, correlated with consumption of pistachios, Consonella with pecan sesame and soy, and Megasphaera with many of these foods. So I think overall what this means is it's not just the environmental exposures, but also on top of that, you have to put on what are the kids eating that will support the growth of specific microbes as they're being acquired. And we think that this is perhaps the overall, the most important effects can be mediated on the immune system via this interaction between foods and microbes. And the way that we think about it is, so here we have a specific microbe, a blue microbe. It likes avocado. It generates this metabolite that has an effect on T regulatory cells. Another microbe, a green microbe, likes a different food, let's say corn, generates a different metabolite. And this metabolite has activity on dendritic cells. And you can start to build up this model, different microbes like different foods, and have different effects on the immune system. But overall, you can see that all these different foods and all these different microbes generate multiple different metabolites that have very dramatic effects on all aspects of immune function. And this is quite a complicated story. There's about 2,000 different biosynthetic gene clusters within the gut microbiome that we know of, many of which can generate some of these metabolites that can have effects on the immune system. But I think also this model is very useful to understand what happens when you take something out. So this microbe over here on the right, it likes coriander, generates this metabolite that inhibits Th2 cells. If you don't eat coriander, then you don't have the substrate, even if you have the microbe. And now this brake on Th2 development has been removed. But the opposite is also true. So if you can eat the food, but you haven't acquired the microbe, you still have this immune deficit. So overall, that gives us a framework to think about how important different diversity of foods and a diversity of microbes is very important in maintaining correct immune function. And this is a review that we published just a few weeks ago, where we looked at, in the lungs, in the skin, and in the gut. And there are many different metabolites coming from microbes that can have effects on pathways associated with asthma, atopic dermatitis, and food allergy. And I'll go through a couple of them in a bit more detail in a moment. So I mentioned G protein coupled receptors earlier, that these were very differentially regulated in rural and urban children. And as I said, these are transmembrane receptors that can couple to different second messenger signaling molecules, that even with the same receptor, you can get multiple different types of responses within the cell. And so we looked in published metagenomic data sets for the presence of genes that can encode for GPCR ligands that would come from bacteria. So red here just means present and blue means absent. But you can see in data sets from over 1,300 individuals for gut samples, 122 airway samples, and 283 skin samples, about 60% of the ligands are present at all body sites. So that suggests to me that these are really important for immune function and immune regulation at multiple body sites. Some are gut restricted and some are skin restricted. And we did look at the skin restricted ones in just a little bit more detail. And in 2 different studies that looked at healthy versus skin, atopic dermatitis skin, there were a number of GPCR ligands, or the genes encoding for bacterial GPCR ligands, that were significantly different between these 2 independent studies. And there was another group that were different either in one study or the other. And we're obviously doing a lot of work with these metabolites right now to see what their influence is on barrier and Th2 polarization. But I guess one that we have done a bit more work on is histamine. So we went looking for bacteria, because histamine is a biogenic amine that you all know is important for allergic responses. And the histamine receptors are G protein coupled receptors. So we were looking for histamine producing microbes from the gut and the skin. And this is a typical platform 1 microbe that we know makes a lot of histamine. You can see here and actually if we put 10 grams of histidine, the amino acid that histamine is generated from, into the culture media, we used to get 10 grams of histamine the next day. So this particular microbe is very good at generating histamine. And we're also able to show that this bacterial-derived histamine activates the human histamine receptors. So then we wanted to know if the bacterial-derived histamine could be having an effect on the immune system. So the allergic responses are typically mediated via the histamine receptor 1 and 4, but the histamine receptor 2 is an immunoregulatory receptor. So we were a bit more interested in that one. And what we did here was in wild type mice, we fed them a lactobacillus that we knew was secreting a large amount of histamine. And when we looked in the mucosa at IL-17 secretion, you can see there's a decrease in the mice that consumed this lactobacillus. But if we did the same experiment in histamine 2 receptor knockout mice, you see the opposite response. And that was also true if we look at other cytokines like IL-4. So our interpretation here is that the histamine coming from microbes in the gut, can have an immunoregulatory effect via the histamine 2 receptor. So in the human gut, Morganella morgani was one of the highest producers of histamine that we found. And actually, when we went back and looked in some of our other data sets, we could see in patients with asthma, there's a higher level of this bacteria in their gut microbiota. And it correlates really nicely with disease severity. So these are patients, severe patients who are not responding to corticosteroids. And I wonder if some of the histamine that's been made by the microbes in the gut may be contributing to symptomology in these individuals. And we wanted to formally test that in a mouse model. So we have here, this is an ovalbumin-induced respiratory inflammation model. And if you look at the inflammatory cells in the lungs, in the positive control, there's a lot there. When we feed the mice an E. coli or an E. coli that's now secreting histamine, so we cloned in the histidine decarboxylase gene into this E. coli, you can see there's less actually inflammatory cells coming into the lungs when the mice are consuming a histamine secreting microbe. And if we look at cytokine levels in the bowels of these lungs, so this is the positive control here, so everything is red. And if you look over here, when the mice get a microbe that's secreting histamine, basically everything is a lot more blue or less red. So there's a very significant effect of histamine that's been made in the gut by a microbe in the gut on distant sites such as the lung. And I guess just a final example is our short chain fatty acids. These are well described to activate G protein coupled receptors. There are many pathways and many different microbes that can make short chain fatty acids, and they generate short chain fatty acids following fermentation of non-digestible oligosaccharides or fibers in the diet. And the three that we most commonly talk about are acetate, propionate, and butyrate. And they don't bind equally to different G protein coupled receptors. So butyrate binds to GPR109A, which isn't bound by propionate or acetate, whereas acetate has a preference for GPR43. And the importance to allergic responses is that these GPRs are expressed by nearly every immune cell that we've looked at. They're important for epithelial barrier function, for mast cell degranulation, for T regulatory cell induction, B regulatory cell induction. So they're really important for immune regulation. In this study, we quantified the short chain fatty acid levels in fecal samples of 1-year-old children. And then we looked at their sensitization to allergens by 6 years of age. And the way that we divided this was those kids with the highest levels versus the lowest levels. So the kids with the highest levels of butyrate at 1 year of age, 26% of them were sensitized to a food or aeroallergen by 6. Whereas in those kids that had the lowest levels of butyrate, 56% of them were sensitized. So a very significant difference. And that's true for both butyrate and propionate. And if we look at some of the disease outcomes, what we can see is, again, if we divide those kids between those with the highest versus the lowest levels, asthma was reduced by 50% in those with the highest levels of butyrate. None of the kids had allergic rhinitis. Food allergy was reduced by about 50%, and atopic dermatitis was reduced by about 1/3. So this very nice correlation between higher levels of short chain fatty acids like butyrate and reduced disease outcomes. Butyrate and acetate were not as significant. So, sorry, propionate and acetate were not as significant. So it looks like butyrate may be the most important for protecting against allergic sensitization. So just to summarize this, so the diet in early life is really important. And the diet that was associated with higher levels of short chain fatty acids were consumption of fruits, veg, yogurts, and fish. So we think that the yogurt is providing microbes, and the fruits and veg are providing fibers that the microbes can ferment in vivo to generate these short-chain fatty acids. And we've shown it, and others have shown as well, that these short-chain fatty acids are very important for lymphocyte polarization to a regulatory response. And these then protect against atopic sensitization, asthma, and food allergy. And this doesn't start just with the infant. This may start even earlier during pregnancy. And again, this is Purina's study from a couple of years ago, where that diet that I just talked about, she looked at mums that were consuming high levels of fruit, veg, and yogurt versus mums that were consuming more processed types of foods, and then looked at allergy outcomes in the offspring. And what you can see is that there is a significant decrease in allergy outcomes in those children born to mums who are eating fruits, veg, and yogurt. We think this is a microbiota mechanism. So just to come to the end, why is it that whole plant foods, or fruits and veg, seeds, nuts, what's so important about them for the microbiota? So there's a couple of different things. So whole plant foods, these are complex food matrices, so they're not easily digestible. So the nutrients that are in the foods will not be fully digested by the host, and a lot of the nutrients will actually get into the colon to feed the microbiota. There's also a lot of dietary fibers, and these are important for production of metabolites like short-chain fatty acids. On the other hand, the heavily processed foods, these are typically acellular, so the nutrition is taken out of the cells are disrupted, so very easy to digest and absorb. So then this reduces the substrate availability for microbes in the gut. There's also certain components of heavily processed foods that may directly damage the microbiota, such as emulsifiers. Typically, dietary fibers are taken out of the processed foods. So again, you restrict the substrates that microbes need for metabolism. And also you can have other components like salt that will directly impact the microbiota. So this is important for chronic diseases, immune-mediated diseases such as atopic dermatitis and food allergy. But just as a last word on this, we also know that this is really important for immune response to infections such as SARS-CoV-2, and we and others have published a lot on this over the last couple of years. And what we're able to describe is that there are some individuals with a low-risk microbiome. What that means is that they have a microbiome that's producing lots of these regulatory metabolites, but very low levels of pro-inflammatory molecules. This results in an immune configuration that when you see something like SARS-CoV-2, you clear the virus very quickly and effectively, and you protect the host from hyperinflammatory responses. On the other hand, there are individuals with a high-risk microbiome, so a very, very different microbiome composition, but also a very different metabolism. So this type of microbiome is not producing the correct levels of the regulatory molecules that the immune system needs, but actually instead is producing a lot of pro-inflammatory molecules. This results in a very different type of immune polarization. And then when these people see SARS-CoV-2, there's delayed viral clearance and consequently the hyperinflammatory responses that damage tissues. And a lot of these differences in the microbiome composition we can see are driven by long-term dietary habits. So finally, to come back to food allergy and to summarize, I think the way we're thinking about food allergy is that there is a low risk and a high risk configuration that is largely determined by the microbiome composition, microbiome metabolism, and the diet of the individual. And so overall, we think that it's the nutritional, environmental, and lifestyle factors that really underpin the immune dysfunction and epithelial barrier disruption that you see in atopic dermatitis and in food allergy. And many of these effects, probably not all, but many of these effects are mediated via microbial metabolism in the gut and on the skin. So I'd like to thank my many collaborators who have contributed over the last few years to the studies that I just shared with you. And I'm happy to be able to join you live in a few moments to answer any questions.